Hi, this is Dr. Lauren Launen, and this is part 3 of an online lecture series intended to introduce you to the biology of the microbe Vibrio vulnificus. In the previous portions of this online lecture series, we introduced you to microbes, and I talked about the genus Vibrio. Now I want to focus in on the specific species Vibrio vulnificus, which is the focus for this project and the work that I do in my lab currently. So we're going to focus on Vibrio vulnificus. This is the genus name, and then this is the species adjective. So the work that we're going to be doing and that you're helping out with is analyzing the genomes of Vibrio vulnificus isolated from the Great Bay Estuary. There has been quite a bit of comparative genomic analysis of Vibrio vulnificus, and one of the things that we know about this organism is that within this species you can organize Vibrio vulnificus strains or isolates, which means the same thing, into 3 different biotypes. The biotypes can be distinguished by looking for the presence or absence of certain alleles, and also by looking at how these organisms behave in the environment and potentially in human hosts. So the 3 biotypes are simply called biotype 1, 2, and 3. Relatively recent whole genome analysis out of the group of Coden, and the references given there, show that the core genome that is shared between these 3 biotypes includes 3,638 genes. That means biotype 1 has 405 unique genes, biotype 3 has 575 unique genes, and biotype 2 has 442 unique genes. Most human infections are caused by biotype 1, and this is in fact the predominant form or biotype that is found in the natural environment. Biotype 2 seems to be mainly associated with eels, and then biotype 3 is a relatively new biotype identified when humans were infected after handling farmed tilapia fish in Israel. So far we think that biotype 3 is somewhat geographically isolated, although future studies will really tell us whether that is true or not. So one of the points that I want you to think about here as you're looking at this slide is the fact that all of these are one single species, but each of these has quite a few genes unique only to that particular biotype. So this should remind you that there can be a lot of diversity, genetic diversity or difference within a single species of microbe. So one of the reasons that we're interested in biotyping is that physiological or phenotypic studies have shown us that representatives from the 3 different biotypes known in Vibrio vulnificus can grow at very different rates. So these are growth curves shown here on the left for biotype 1, 2, and 3. And what this is it's looking at a measure of cell number over time, and you can see from the shape, it's not a linear shape, so it's showing us the exponential phase of cell growth. And what I want you to note is that this curve is steepest for biotype 1, steeper than for biotype 2 or biotype 3. So if we were to rank the isolates studied and shown here, we would say that the growth rate is fastest for biotype 1, next biotype 2, and lastly biotype 3. Now one of the things that modern molecular microbiological studies are showing us is that growth rate is genetically determined. And we also know from medical microbiology that growth rate is an important virulence factor. So many pathogens, one of their characteristics is that they can grow very, very quickly. So we know that most pathogenic Vibrio vulnificus are biotype 1, and perhaps not surprisingly, it seems like biotype 1 has the fastest growth rate in culture and likely also in the human body. Now that is something that we are starting to be able to study genetically, to look for alleles associated with fast growth rate, and that's going to actually be a future direction for our project, linking the genetics and the phenotype and growing isolates in the lab and measuring their growth rates, comparing them with known pathogens. The genetic typing of Vibrio vulnificus is fairly complex, and there's a lot of history to it. So I mentioned the 3 biotypes, which can be distinguished physiologically and genetically, But there's an additional way of genetically categorizing Vibrio vulnificus, and that's to categorize it as either clinical or environmental using genetic analysis. So C here means clinical, and E means environmental. And genetic studies that were published, again, by Coten et al. In the paper that I mentioned previously have shown that clinical and environmental isolates do group somewhat differently. So down here we have biotype 1 isolates of Vibrio vulnificus that all cause human disease, and they are all biotype 1, and they're grouping with a clade that is being referred to as clinical. Here we have another clade. It's the environmental type 2 clade, and it includes human pathogens as well, and they're all of biotype 3, and that's confined to infections associated with farm tilapia in Israel. And then up here we have the majority of what we find in the environment. These are not pathogenic strains, mostly biotype 1, a little bit of biotype 2, and these groups separately. So the point I'm trying to make here is that there are clear genetic differences between those Vibrio vulnificus that cause disease and those that don't. And we can determine this through genetic analysis. So let's say that we're focusing in on the pathogenicity of Vibrio vulnificus. Well, one of the things that we know is that in order to catch a pathogen, you have to be exposed to the pathogen. So how does that happen? Vibrio can infect the body in two ways. They can infect the body because you're exposed through ingestion. So here with Vibrio vulnificus, that would be by ingestion of contaminated seafood, most commonly raw oysters. Or your exposure could be dermal. Dermal means through the skin. And typically that means that you had some kind of an open wound, and that could be a very, very small cut or scrape or scratch. And that open wound is exposed to contaminated seawater or seafood. So we see this happening in people who have been swimming or in fishermen or oyster harvesters. The pathogenicity of Vibrio vulnificus has received attention in the last couple of years. This is a slide from the summer of 2015 when there were a couple of pretty terrible Vibrio vulnificus infections associated with swimming, where swimmers were exposed to contaminated seawater, and they must have had a cut, and that Vibrio vulnificus entered the cut, and the infection that developed is a kind of necrotic fasciitis, otherwise known as flesh-eating disease, and that's associated with a really high mortality rate. So this is a pretty dramatic and terrible illness. In general, we can categorize illness from Vibrio vulnificus in three ways. Vibrio vulnificus can cause gastroenteritis, and that means you've eaten the organism and the symptoms are vomiting, diarrhea, and abdominal cramps. Not a high fatality or mortality rate with that. Or you could develop severe or primary septicemia. This is usually from ingestion, but it can also be associated with wound exposure. This is a much, much more serious kind of illness due to this pathogen. You'll have fevers, chills, vomiting, nausea, diarrhea. You can see very painful skin lesions like the ones shown here. This has a 50% mortality rate, so it's very, very serious. And we can also have illness that just shows as soft tissue or skin infections, usually due to surface exposure. These might resolve quickly, or they might also be followed up and get worse and become a form of septicemia and then be associated with 50% mortality. It's worth mentioning wound infections as a particular category of illness, or like sort of one type of illness, and you can have wound infections from V. vulnificus when skin lacerations or abrasions come into contact with the organism. You'll see redness, swelling, and intense pain, fluid-filled blisters, and you can get tissue necrosis or death. It can look like an illness called gas gangrene, and this is usually going to have to be resolved by amputation or surgical debridement. Very high mortality, and we see about a quarter of the cases of all Vibrio vulnificus illnesses are due to wound infection. It's a very serious concern. So the people who are most at risk due to infection, or most at risk of infection when they are exposed to Vibrio vulnificus, have generally underlying health conditions. So there are people who have liver disorders like cirrhosis from alcoholism, or liver cancer, or other forms of hepatitis. They are people with a disease called hemochromatosis, which causes iron overload in the liver, people who have diabetes mellitus, and specifically where the diabetes is not well controlled, people who are already sick with something that suppresses the immune system like HIV/AIDS, or they're getting treated for cancer, and also individuals who take prescribed medication that lower stomach acidity levels in a chronic manner. They are particularly at risk of developing infection. If any of these conditions apply to you, then you are 80 times more likely to be infected if exposed. For the rest of us, if we're exposed, we generally won't become infected, so there won't really be any way of knowing that you were exposed. The incidence of Vibrio vulnificus in the United States, there are about 95 or 100 cases per year that are confirmed illnesses due to Vibrio vulnificus. About 85 of these result in hospitalization, and we see around 35 deaths per year. Most of these infections occur in people who have been in the southern states, the southern coastal states, so Mississippi, Alabama, Louisiana, Texas, or especially Florida. So statistically, this is a gruesome illness to get, and you are more likely to get it than to be eaten by a shark. So I would worry more about eating raw oysters than shark attack if I were vacationing in Florida in the warm summer months. There is a seasonality to Vibrio vulnificus infections. So this is a graph taken from a paper in Japan, and what we're seeing in the pink bars are number of sick patients. And in the blue line, we're seeing seawater temperature. And so you can see that in the parts of the year where seawater is very warm, that's when you see infections. When seawater is cool, we see 0 to no infections. And so what we see in the United States is Vibrio infections that occur basically in the warm summer months. Here we're in a different hemisphere. So again, just note that it is infections associated with warm summer months. So why don't you get Vibrio infections in the wintertime? And that's a big question, a big area of research. And the dominant theory right now, and there's plenty of evidence to support this, is that Vibrio are one of many kinds of bacteria that can go into a dormant or stressed state where they basically are hiding out and they're completely metabolically inactive. When you are exposed to organisms when they're in that state, and that state is referred to as viable but not culturable, or VBNC, then the organism is just like an inert particle and it won't infect you. And it seems like cold temperatures trigger the bacteria to go into the viable but not culturable state and just hang out in sediments over the winter. So we can't recover them in monitoring programs, and we also don't get sick from them when we're exposed to water or oysters collected during those cold winter months. So we're primarily exposed by ingestion or through water use like swimming or fishing, if you consider that a water use. That's how we're exposed to this pathogen. Whether or not we are exposed to high levels of the pathogen depend on environmental conditions. So for example, how warm was it on the boat where the oysters were sitting after they were harvested, and did that give the microbe the time to be metabolically active and to multiply? What's the phytoplankton load? So are the oysters taking up a lot of the phytoplankton or the zooplankton load? Are they taking up a lot of these bacteria due to filter feeding? Because the bacteria are found in higher levels in waters that are warm. What's the temperature, what's the pH, and what's the salinity? All of these things affect the levels of Vibrio vulnificus, both in the water, in the oysters, and under harvest conditions. So there's a lot that goes into the management of oyster fisheries and beach management to protect us from Vibrio exposure, either Vibrio vulnificus or other types of Vibrio, or even other types of microbe. And that's part of the system that information on Vibrio vulnificus that we gather through our project will help us to feed into.